Are there mermaids? Can a centaur win at the Olympics? Would you vote to test drugs on human-animal hybrids? Has a long-held dream of scientists finally come true? Will our future be populated by new species developed in the lab? How many can we create, and for what purpose? In 2018, Chinese scientists created the first genetically engineered baby. And before that, rhesus monkeys with a human brain gene associated with intelligence. Russia followed soon, while Stanford geneticists injected human brain stem cells into mouse fetuses back in 2005 to create mice whose brains are about 1% human. U.S. scientist Juan Carlos Ispizua Belmonte, who manages a lab at the Salk Institute in California, and his team created the world's first human-monkey hybrid embryo in China to avoid legal problems in the United States. Japan lifted its ban on creating animal embryos with human cells transplanted into surrogate animals in 2019. The British scientists created the hybrids in secret in 3 laboratories at King's College London, Newcastle College, and Warwick College. If you want to know what's going on here, and if our world is quietly adopting Dr. Mengele's paradigm, watch the video to the end and leave us a comment anytime. Do not forget to share and like the video to spark discussion about this controversial practice on the edge of ethics and innovation. Subscribe to our channel for more amazing facts and stories. It has been said that given a choice between tradition and innovation, society will always choose innovation, just as nature does. The question is whether man can control nature or whether he is part of nature and therefore controlled himself. Let's begin with the theory. What if the mythical creatures, half man, half animal, actually existed in a perished civilization that was wiped out by a cosmic natural catastrophe, caused by an asteroid or the escape of water from Earth's atmosphere as a result of Venus's violent entry into the solar system? This is just a theory in the spirit of the exciting Earth changes our civilization is currently experiencing. A civilization before the time of the Egyptians, perhaps Atlantis? Already Plato wrote about Atlantis. You should check it out if Atlantis speaks to you. And what if these half-human, half-animal creatures were genetically created by the advanced society of Atlantis to serve as slaves to humans? I don't want to scare you and say that our civilization has also reached this point where we genetically mix humans and animals to create a new creature that certainly has no human rights and is used by society to exploit its engineered yet individual nature. But the fact is that we are creating something that is not the natural order, and we have been doing that since the 1920s in the US and even earlier in Russia. Since 1910, long before the Nazis put their hands in the inspirational gene pool. So let us delve deeply into the matter. A chimera is an organism with genetic material from 2 or more sources. Human-animal hybrids, also called chimeras, are living organisms with a mixture of human and animal cells. They are created by injecting human stem cells into the embryo of another animal. Hybrids are named by the convention first part of the father's name plus second part of the mother's name, unless the result is unwieldy. Thus, human means a cross of male chimpanzee and female human, while human-zee or man-pansy means a cross of male human and female chimpanzee. In the past, human-animal chimeras were unattainable. Such experiments aren't currently eligible for funding in the United States. Nevertheless, in 2017, a team led by Juan Carlos Ispizua Belmonte at the Salk Institute in California, with private funding, attempted to grow the first embryos from cells of humans and pigs after they succeeded in mixing mice and rats when they used the genome editing tool CRISPR to hack into mouse blastocysts, the precursors to embryos. There, they deleted genes the mice needed to grow certain organs. When they introduced stem cells from rats capable of producing those organs, those cells thrived. Most of the mice managed to live to adulthood. Some even grew chimeric gallbladders from mouse and rat cells, even though rats don't have this organ. However, the 2017 pig-human experiment, in which the team took stem cells from rats and injected them into pig blastocysts, wasn't as successful. Of 2,075 implanted embryos, only 186 developed within the 28-day project period. This was no surprise, because rats and pigs have completely different gestation periods and evolutionary antecedents. But pigs also bear a remarkable resemblance to humans. Although they take less time to gestate, their organs look very similar to ours. Still, human tissue seems to slow the growth of the embryo, and organs grown from such embryos as are now developing would probably be rejected by humans because they would contain so much pig tissue. Meanwhile, across the ocean, Portuguese researchers created a chimera virus, a mouse virus with a human viral gene, which allowed them to explore new ways to treat cancer caused by infection with the human herpes virus. The team found that when the cancer-causing virus loses a protein called LANA, it also loses its ability to cause cancer. The following year, Isbisua Belmonte and his colleagues, in collaboration with Weizhi's group at Kunming College of Science and Technology in Yunnan, China, have produced human-monkey chimeras. Because our evolutionary relationship with monkeys, they used macaques, is closer. We only separated about 20 to 30 million years ago. The researchers thought the two cell types might get along better and hoped to find out more about what promotes their compatibility. And what did they do it? They fertilized squirrel monkey, Macaca fascicularis, eggs and grew them in cultures. 6 days after fertilization, the team injected 132 embryos with human-expanded pluripotent stem cells, which can develop into a range of cell types inside and outside an embryo. The embryos each developed unique combinations of human and monkey cells and deteriorated at different rates. 11 days after fertilization, 91 embryos were alive. By day 17, only 12 remained, and by day 19, only 3 remained. According to the researchers, the human cells generally seem to integrate better into the monkey cells than into pig embryos. Several embryos still contained about 4 to 7% human cells on day 15. This means that human-monkey chimeras would allow closer and more extensive mixing of the two cell types, with less predictability about where they will eventually grow. These chimeras could then be used as experimental subjects for research into human development and disease or for testing drugs. They could even be used to grow human organs for transplantation. But, a big but, the team, like others in the past, was unable to control which cells develop into which tissue, which is an important step to master before such models can be used. At this point, scientists agree not to make human-animal chimeras with a high human content, and instead use genetically modified human stem cells that cannot form brain cells. This is because with more intelligent creatures that can understand their condition, the ethical concerns increase immeasurably. The International Society for Stem Cell Research, ISSCR, currently prohibits researchers from allowing human-animal crosses and recommends additional monitoring if human cells could integrate into the developing central nervous system of an animal host. Many countries, including the United States, Canada, the United Kingdom, and Japan, have temporarily restricted research on chimeras using human cells. Japan lifted its ban on experiments with animal embryos containing human cells in 2019 and began funding such work. In 2015, the U.S. National Institutes of Health, NIH, announced a moratorium on federal funding for studies that inject human cells into animal embryos. In 2016, the funding agency proposed lifting the ban, but limiting research to hybrids created after gastrulation, when the early nervous system begins to form. More than 6 years later, the funding ban is still in place. The question is how much social control there should be over the direction science takes, and we know that science has always challenged social norms. Russian biologist Ilya Ivanovich Ivanov made the same attempt in the 1920s under the auspices of the Red Army, but could not achieve a successful pregnancy in female chimpanzees artificially inseminated with human sperm. Joseph Stalin was a staunch Darwinist and funded the program to create super warriors from ape men. He reportedly said to Ivanov, "I want a new, invincible man, insensitive to pain, resistant and indifferent to the quality of the food he eats." Ivanov only tried to impregnate 3 females before he had to abandon the project as useless. Despairing of his limited financial resources, Ivanov made the terrible decision to attempt the impregnation of African females with chimpanzee sperm without their knowledge. He submitted a proposal for his experiment to doctors at a local hospital in Kindia, French Guinea, and was ready to go ahead with it when the Governor General of French Guinea, Paul Poiret, rejected the plan. Ivanov hoped to continue his experiment in Russia with the help of women volunteers, and he found at least one willing to participate. However, when it became known that Ivanov had attempted to inseminate African women without their consent, he was condemned by the Soviet Academy of Sciences and all support was withdrawn. Stalin was convinced at this point that genetic research would lead to eugenics and fascism, and stopped all projects. He killed most of the scientists as enemies of the people and threw others into forced labor in Siberian gulags. On December 13, 1930, Ivanov was arrested by the secret police and convicted of founding a counter-revolutionary organization among agricultural specialists, and exiled to Kazakhstan, where he died 2 years later. His chief accuser took Ivanov's position as head of the laboratory. The question of where to draw the line between a human and a non-human being is troubling to many researchers. While animals whose cells are 1% or less derived from humans clearly seem to be in the same boat as other animals, there is no consensus on how to think about beings in the genetic middle ground that exhibit something like an even mix. Drop us your thoughts in the comments and do not forget to like and share the video. We hope we have opened a can of worms, in a good way. Stay tuned or subscribe now and be the first to hear about our amazing stories.